So what we've just done is that we've listed several criteria, three criteria, that would give a gene region a lot of potential as far as possibly influencing smoking behavior. So what we're going to be asked to do is apply that information. So again, here's a picture of the dopamine pathway and a lot of the molecules that are involved with that pathway. So what we're going to be asked to do is consider the genes that encode for each one of these molecules. Not only that, we're going to be asked to consider specific regions in each one of these genes and see if we can't identify the specific regions that might have the best potential as far as influencing smoking behavior. So to see how that's done, how about if we consider the gene that encodes for this dopamine receptor right here. And so here's a picture of the gene that encodes for that particular dopamine receptor. So this is a type of dopamine receptor called the DRD2 receptor. And so this picture has a lot of information. So if you just break it down, it might make more sense. So again, this whole thing shows the entire gene basically for the DRD2 dopamine receptor. Here is the promoter for that gene. The terminator for the gene is somewhere to the right here. It's not shown in this picture, but if you go way out to the right, you probably would expect to see the terminator out there somewhere. Remember we just said that if you're talking about a eukaryotic gene, there are several exons within it, and there are several introns within it. And so that's what all of these E regions represent. So E1, E2, E3, E4, those are all exons within this particular gene. All of these I regions, I1, I2, I3, I4, those are all introns within this particular gene. So all of these white spaces would be taken out before this gene ends up being translated. What this means here, so these dashed lines just mean that there's a portion of the gene here, actually 250 kilobase pairs worth of gene, that's actually not being shown in this picture because it's not relevant to what this picture is trying to illustrate. Same thing here, this is saying that there's just a big chunk of gene here, 10 kilobase pairs worth, that's actually not being shown because it's not relevant to what this picture is trying to illustrate. There's a few other things that are being shown in this picture. So you see that there are some arrows indicating where different single nucleotide polymorphisms exist within this gene. And so if you remember how we define single nucleotide polymorphism, basically what this is saying is that there is a particular nucleotide in this area of the gene that tends to differ between different people. Same thing here, this area, this area, this area, and this area contain nucleotides that tend to differ among different people. And so again, here's what the DRD2 gene looks like. In your assignment, you're given a table that kind of gives you information on different portions of this gene. So here's what that table looks like. Right, so again, this describes, so what this left-hand column shows is it lists different regions within this gene. And then all of these right-hand columns give descriptions of each one of those regions. So for example, this box here says that there's a part of the DRD2 gene called the -141C insertion deletion. And actually in this picture here, it points out where that region is. The description of that particular region, it says it's an insertion deletion of cytosine in the promoter 141 bases before the start of transcription. So that makes sense, right? Because that region, it's being shown that it's in the promoter, and because it's within the promoter, it's before where the first nucleotide is that the first nucleotide of this gene. It's before that, which would be around here somewhere. All right. It says that furthermore, for the description, it says it involves an insertion deletion of cytosine in that promoter. So basically, what that's saying is this: in some people, if you consider this particular region, their gene in this region contains the nucleotide cytosine, whereas in other people, that cytosine or that cytosine is not there, it's deleted. So that's what 141C insertion deletion means. 141 tells you the position, it's minus 141 base pairs from the first nucleotide. C insertion deletion tells you that in some people there's a C in that region, whereas in other people there is not a C in that region. It tells you more stuff here about this particular region of the DRD2 gene. It tells you that what percentage of European Americans have the deletion in that particular area, what percentage of African Americans and Japanese have the deletion in that particular area. It tells you here that having the deletion in that particular area causes this. So cells that have that deletion resulted in lower transcription compared to the insertion. So by having that missing C, it causes that gene to make less proteins as compared to a gene that does contain that particular C. And then so here are some associated addictive behaviors with that particular deletion. And so if you are given this information, then you might be able to figure out or judge, is this a good region to study? In other words, does this region have a lot of potential in terms of something that might cause a difference in smoking behavior? And so you'll be asked to kind of write that out in your assignment in this particular table. So what this is saying is this. Consider this region of the DRD2 gene, the one we just looked at. It's asking you, based on what you know, might this be a good candidate as far as a gene region that causes differences in smoking behavior? In this particular case, I think there's enough information here to say that this is probably a good candidate based on those criteria that we listed before. Specifically, here are the reasons why. This polymorphism that we're looking at here, it occurs in a region of the genome, specifically the promoter, that actually causes a difference in how much gene is transcribed. Right? So here in this table, it's saying that if there's a deletion in this particular part of the gene, it actually has a biological effect. Right? It causes less protein to be produced. That was one of the criteria that we listed as far as something that might make a gene region a good candidate. Not only that, but this polymorphism, it occurs at a relatively high frequency in different ethnic groups. So again, this portion of the table here tells you that it's, again, a relatively high frequency between different ethnic groups. And that was one of the criteria that we listed as far as what might make a gene region have a lot of potential as far as possibly causing differences between different people. Right? So by using the criteria that we listed before, and by using the information in tables like this, you should be able to make an argument as far as whether a gene region is a good candidate to study or a bad candidate to study as far as possibly having an effect on smoking behavior. And so you're going to be asked to make these judgments for several different gene regions. And so here's a more complete look at the table that kind of breaks down each polymorphism within the DRD2 gene. You're also provided with a table that talks about different polymorphisms in a DDC gene and a DAT gene. So there's several different gene regions that we could possibly look at, and you're going to be asked to evaluate each one as far as its potential for causing differences in smoking behavior.